My name is Taylor Lenz, and the project I've been researching and looking into over the past couple of weeks is the metagenomics of the human intestine gut microbiota stability and fluctuation. This has always seemed really interesting to me due to the fact of how many different microbiota there are within the human and how that plays into different areas of the body and optimal health and function. And some of those different things we'll look into throughout the rest of this presentation. There are many small, complex, and diverse microbiomes throughout the human organism, each specializing in the functionality and health of the various parts of the body. One such microbiota is located in the human intestinal tract. This microbiota is associated with the preservation and repair of the digestional tract from beginning to end, and is comparable to the function of a human organ. The interconnections of the human gut microbiome are complex, and when the delicate balance is disrupted, it decreases optimal health and performance. So the thesis statement for this project is the interconnections of the human gut microbiome are complex, and when the delicate balance is disrupted, it decreases optimal health and performance. Because of the function of an additional human organ, such as this microbiome, it's imperative to the health and life the healthy life and function of the internal systems. The balance of the microbiome is critical to the quality of life and the health of an individual. The intestinal microbiome not only determines the function of the digestive tract, but also radiates efficiency and performance to the other systems in the organism due to the interconnections of the complex scheme of systems in the human body. So, in the balance of the microbiome, a few of the essential functions of a balanced microbiome include a healthy metabolism of nutrients, a functioning gastrointestinal and bowel system, and an operative immune system. The balanced intestinal tract is done so by maintaining regularity within the diet of the individual and live culture consumption through products such as kombucha or yogurt. And this balance can be pivotal to the lifespan and performance of the individual and is present from birth to death. So in the imbalance of the microbiome, imbalances of the microbiome can come from a multitude of sources, including a drastic diet change, heavy antibiotics, and other harsh medicines. Disturbances such as this can cause decreased health of the individual and poor digestion performance. Some of the more severe implications of a microbe imbalance consists of obesity, inflammatory bowel disease, pulmonary diseases, and urinary tract infections. Because of these implications, a microbiome offset, it is imperative to strive for an establishment of stabilized microbiomes. What the main staple of this ever-changing system is the fluidity of it. Each individual has a very unique set of microorganisms making up the microbiota, and through the change in development of the individual, such as in the consumption pattern alterations and illness requiring medicine usage, the microbiome changes as well. And through these factors, as well as natural evolution through the microbiome. This evolution in the microbiome, as seen in these photos here, the changes are how it relates to genetics and how even though each system is unique to the individual, those microorganisms specifically are also incredibly unique. And there's a form fits function, and where the evolution changes throughout that organism, its role in the microbiome changes. So how does this relate to genetics? The way that the human intestinal microbiome plays into genetics is in the way that each individual microbe has its own set of genes, and with the rapid evolutionary changes taking place in the species of the microbes, causes for different forms and functions of the organism, making an effect on the host individual. Through metagenomics and the exploration of the human gut microbiota, specific microbes can be evaluated and studied, showing each of the functions as and which genes are pivotal in the functionality of the overall system. The study of this can be done through pure culture studies in the communities living within those cultures, which can be done across species. And of course, these practices also have their own set of restraints and setbacks and negatives, such as not all bacteria can be cultured using the same methods, not all are across species, only some are particular to certain species and things of that nature. Overall, the metagenomics of the human gut microbiota is ever-changing, and as the principles of evolution express, those changes are affecting the health and function of the human individual. These changes affect the stability and fluctuation of the microbiota, and human intervention via personalized genomics and medicine are only a few of the ways in which maintenance and care are established, making it one of the ever-growing fields of metagenomics. Oops. Through the study of particular cultures of a given species, including the human, through the study of those cultures, specific microorganisms can be targeted, and then not only that, but the genes of that particular microorganism can then be targeted and dissected and looked at in a way to see which gene helps regulate and promote the function of the different role of that particular microorganism in the overall microbiota of the culture. And that particularly is what makes it such an interesting topic to look into for metagenomics. The evolution of these microorganisms are ever changing, as it is with any organism, but because of the interconnections in the system in which they operate and run a organism on the whole and can be considered that additional organ of the individual, it makes the entire system more complicated and more delicate, and which makes that balance versus imbalance that much more important to recognize. To end, this is my list of sources that I utilized for this presentation. There were so many that I came across that could have been used. This field is so interesting when it comes to personalized genomics and medicine and personal health when it comes to the microbiota. And there's so much more to learn about it. I didn't include nearly a portion of everything that could be found. And I would really like to continue a research project in a few more years to see where it's grown since now, since the personalization of medicine and everything has already become so particular, to see where science could take us in the metagenomics field related to the microbiota of the human organism. Thank you all so much, and I hope you found it as interesting as I did.